Hi, my name is Dongjoon Lim, and I'm a PhD student at McGill University, supervised by Professor Matthew Blanchet. I will be presenting our paper EvolSTM, Context-Dependent Models of Sequence Evolution using a sequence-to-sequence LSTM. Models of sequence evolution describe the rate at which mutations take place. Many of the most fundamental tasks in bioinformatics rely on these types of models, including pairwise and multiple sequence alignment, phylogenetic inference, ancestral genome reconstruction, and simulations of neutral sequence evolution. In our project, we aim to develop machine learning approaches to model sequence evolution, and to do so in a manner that considers the impact of the sequence context. Many early but commonly used sequence evolution models make oversimplifying assumptions about mutational processes. For example, the Jukes-Cantor or Kimura's model of substitution, as well as the linear or affine gap penalty scoring scheme, assumes mutation rates to be constant and independent of the sequence context. However, mutation rates are known to have strong context dependencies. For example, the probability of cytosine mutating to thymine is elevated in the context of CpG dinucleotide caused by the deamination of a methylated cytosine. Also, it is known that the probability of insertions and deletions is elevated in locally repetitive sequences due to the DNA polymerase slippage. Thus, using context-independent evolution models could result in inaccurate alignments, inability to detect remote homology, incorrect phylogenetic inference, unrealistic sequence evolution simulations, and so on. Several approaches have been proposed to model substitution and indel rates in a context-dependent manner. Early approaches were based on simple word counting analyses, whereas more recent approaches take more sophisticated statistical approaches. However, due to the exponential blow-up of computational costs with the context size, these methods often limit the context up to 2 flanking bases around the mutation site of interest, which means there is a need for an approach to capture longer-range context dependencies. And to achieve this, we could think about using new machine learning approaches to solve this problem. To overcome the limitations of previous models, we introduced EvolSTM, which is an evolutionary model that takes advantage of machine learning approaches developed in the context of natural language processing. More specifically, EvolSTM is based on a sequence-to-sequence model using long short-term memory, or LSTM, to take advantage of the very large quantities of genomic sequence alignment dataset to train the model on. This is a brief overview of our approach. To train EvolSTM, we first extract a training dataset in the form of a large number of aligned k-mers for some ancestor-descendant pair. We then train a sequence-to-sequence model using this dataset, aiming to predict the probability of substitution and indels as accurately as possible. Finally, once a model is trained, we can use it to either simulate the evolution of a given sequence or calculate the likelihood of a given ancestor-descendant pair. Let us look at the detailed steps of extracting the dataset. We used a 100-way alignment of whole vertebrate genomes and applied the Ancestors 1.0 program to infer their maximum likelihood ancestral sequences. We then extracted the pairwise alignment between the human genome and the Old World Monkey Ancestor genome to train the model, since the reconstruction has been shown to be particularly accurate for primates. Afterwards, the gaps present in both sequences are removed. Deletions will be treated as a special type of substitution, but insertions need to be handled differently. Then each portion of 1 or 2 nucleotides in the descendant sequence aligned to consecutive gaps in the ancestors were combined with the previous descendant nucleotide and represented as a single character from an extended alphabet that contains all mono, di, and trinucleotide sequences. Finally, these modified alignment is sliced into k-mer pairs and are one-hot encoded. We made 5 versions of Dataset with different k-mer sizes, 5, 11, 15, 21, and 39, to later test and identify the optimal context k-mer size. The dataset is then split into the training set with 10 million k-mer blocks and the validation set with 2 million k-mer blocks from the human chromosome 2 portion. The test set consists of nearly 150 million k-mer blocks from all other chromosomes. The size imbalance between the training set and the test set is intentional, so that the very large test set would enable us to accurately estimate the accuracy of the trained model. Now let us look at the model structure. This is an example showing the modeling of 3-mer ACT evolving into AGT. The sequence-to-sequence model is composed of connected LSTM-based encoder and decoder networks. The encoder is shown in red. It is composed of 2 LSTM networks receiving the same input ancestral k-mer, but passes the hidden state and the cell state vector in opposite directions. The loss output hidden state, which is the red arrow, and the cell state, which is a blue arrow, are passed to the decoder network. Each cell state and hidden state is captured using a vector of size 1024, which captures context information that will facilitate the decoding stage. The decoder network, in blue, is trained in a teacher-forcing fashion, where the target metanucleotide is concatenated with the nucleotide in the ancestral k-mer to be passed as the next input. The training goal was to minimize the cross-entropy loss of the prediction. We used Atom Optimizer with a learning rate of 0.0001 to train the batch size of 1024. Once trained, a model can be used to simulate the evolution of a given ancestral sequence. This is an example of simulating a descendant 3-mer given the input ancestral 3-mer ACT. The only difference here is that for the decoder network, the metanucleotide is randomly drawn from the emitted output distribution of probabilities and passed onto the next LSTM decoder cell to predict the metanucleotide in the next time step. To analyze EvolSTM's ability to capture long-range context dependencies, we need a baseline model to compare to. However, past studies focused on immediate neighbor context-dependent mutations, so their approach cannot be compared directly here. Thus, we need to implement a simple context-dependent evolutionary baseline model. For this purpose, we introduced a simple frequency table method for longer-range context-dependent evolution. We first tabulate the probability of observing descendant k-mer D given ancestral k-mer A for the k size from 135 up to 39. Then, to calculate the probability of ancestral k-mer A evolving into descendant k-mer D, we look up the corresponding A and D pair in the table. But when k is large, most ancestor-descendant pairs have 0 observations. In that case, we symmetrically trim A and D by 1 nucleotide at each end, resulting in a k-2-mer alignment, and then we continue trimming until the ancestor-descendant pair has an observed frequency greater than 0. We first evaluated the ability of different approaches to properly estimate mutation probability by comparing the cross-entropy loss of each approach calculated on the same test set. We have trained 4 different approaches, including EvolSTM with 1 layer of LSTM network, EvolSTM with 2 stacked layers of LSTM networks, as well as the simple bidirectional LSTM and the frequency table approach. We can observe that the EvolSTM with 1 layer takes the advantage of the context size up to 15, with the lowest cross-entropy loss achieved among all approaches. The other approaches, as in particular the frequency table approach, are not as successful. Another thing to observe is that the performance of LSTM-based approaches worsens with the context sizes larger than 15. This could be due to the unstable gradient problem of LSTM with large input sequences. So for the rest of the experiment, we used a k-value of 15. We evaluated EvolSTM's ability to capture the context dependency of mutation within two flanking bases on each side of the mutation site. Each graph shows a comparison of frequencies of a given mutation. For example, C to T substitution in the both left corners, occurring at the center of the 5-mer, between the EvolSTM simulation on the test set and the observed test set. The dots in the graph corresponds to the 256 possible 5-mer contexts. We can observe that the Pearson correlation coefficient is high, ranging from 0.83 to 0.99, showing that EvolSTM efficiently captures the context dependency of size 5. Also, EvolSTM was able to capture the elevated probability of C mutating to T and its reverse complement G mutating to A within the context of dinucleotide CpG, showing a clear two clusters of context in the top left and bottom left graph. Moreover, we could also observe that even within the CpG dinucleotide context, the probability of C to T mutation is strongly dependent on the broader context, with a twofold difference in mutation probability between the least and the most mutagenic CpG contexts. We performed a further analysis by testing the effects of adding extra flanking-based context on the most mutagenic context of C to T mutation versus the least mutagenic context of C to T mutation. The ACG context is the most mutagenic trinucleotide context. Then by adding one more context based on each flank, we can observe that there is a 35% difference in C to T mutagenicity between the most mutagenic flanking-based context of ACG, which is CACGG, and the least mutagenic AACGA context. Bringing it further to the context size of 7, there is a twofold difference between high and low mutagenic flanking-based contexts of GACGG. Throughout, the correlation coefficient between the predicted and observed mutation probabilities remain quite high, until the context size considered become less relevant to mutation probability, for example, the flanking-based context of ACCTT. Thus, we can conclude that EvolSTM is able to capture the mutagenicity within a larger context size. To further study the impact of long-range context on mutation probability, we looked at the log ratio of the probability of mutation in a given context and the probability of the same mutation in an extended context. When the log ratio is negative, this means that the extra context on each flank reduces the mutation rate, and when the value is positive, the extra context increases that rate. Here, we're taking advantage of the fact that most mutational processes are invariant with respect to strandedness. So we would expect the log likelihood ratio of a k-mer and its reverse complement to be comparable. This is what these graphs show, with the x-axis showing the log likelihood ratio for a k-mer, and the y-axis showing the log likelihood ratio for the reverse complement of that k-mer. Each dot in the graph corresponds to a configuration of extra context, and these dots are concentrated along the diagonal of the graph, which is consistent with our expectation. However, as larger context sizes are considered, for example the k size of 7, the range of log likelihood ratio values obtained is reduced, and the reproducibility between a k-mer's log likelihood ratio and that of its reverse complement is reduced, showing a reduction in impact on mutagenicity. In conclusion, we observe that EvolSTM efficiently captures local context evolutions, and does so more accurately than approaches based on frequency tables. We observe that certain mutations are strongly context dependent, and that this effect often extends to 3 to 4 positions upstream or downstream. As such, we could expect that EvolSTM will provide a better benchmark in simulating highly diverged sequences. With the ability to model context-dependent evolution, EvolSTM could potentially be used for building a better scoring scheme for sequence alignments and homology detection. However, EvolSTM still has a room for improvements. We have ignored the insertions with length larger than 2, so EvolSTM is not able to model these instances of evolution. Also, some mutational events are not considered. For example, instances of large-scale insertions of transposable elements will not be captured in EvolSTM since the k-mer size it takes is 15. We are currently working on improving EvolSTM's accuracy, efficiency, and scalability. First, many breakthroughs in the field of recurrent neural network have emerged, including the attention mechanisms in sequence-to-sequence model and transformers, mainly utilizing the attention mechanism instead of traditional LSTM or GRU cells. It would be worthwhile to modify EvolSTM to use these cutting-edge machine learning methods. Since we have built a context-dependent evolution model, it would also be worthwhile to ameliorate sequence alignment methods using more accurately calculating scoring schemes. Lastly, we could use EvolSTM for detailed study of context-dependent mutation in different types of genomic context, such as different behavior in coding region and non-coding region. We thank all members of Blanchette Lab, especially Sean McRae, Zichao Yan, and Elliot Lane for the useful comments and suggestions. We also thank Genome Canada, McGill University, and NSERC for financially funding the research, and Compute Canada for providing the computing resources. Thank you for listening.